Well, students, today we are taking the topic gametogenesis. Gametogenesis means formation of gametes in the male and female reproductive organs. Primary male reproductive organs are pair of testes, and in females reproductive organs, primary reproductive organs are pair of ovary. So inside the testes and ovary, sperm and ovum are produced or formed, and that process is called gametogenesis. In males it is called spermatogenesis, and in females it is called oogenesis. So first we take spermatogenesis. Inside the testis there are seminiferous tubules. Seminiferous tubules lining consist of two types of cells, spermatogonia and Sertoli cells. That spermatogonia produce male gamete. Spermatogonia are diploid cells. They undergo mitosis and form primary spermatocytes. So primary spermatocytes are also diploid cells. Primary spermatocytes divide by meiosis first, actual reduction division, and form two haploid secondary spermatocytes, and this division is equational division, and cytokinesis is also equal, because in oogenesis we'll see unequal cytokinesis. Meiosis second in secondary spermatocytes then divide by meiosis 2 and form four haploid cells called spermatids. So spermatids are undifferentiated male gametes, they can be called. Spermatids now transformed into mature sperm cells by a process called spermiogenesis. Which process? Spermiogenesis. So you can see this lining is germinal epithelium, where the spermatogonium cells are there and Sertoli cells are there. Spermatogonium divide by mitosis from primary spermatocytes seen somewhere after the spermatogonia. Primary spermatocytes are dividing into the spermatids, and spermatids are converted into spermatozoa, and conversion of this spermatid into spermatozoa is carried out by Sertoli cells. They provide nutrition to developing sperms. So see this process once again. This is spermatogenesis. Okay, once again see it inside the camera. See in the camera. See, this is spermatogonium diploid cell, divide by mitosis and form two primary spermatocytes. Okay, both are diploid. Each spermatocytes now divide by meiosis first and form first haploid cell called secondary spermatocytes. This is meiosis first. This process all is taking place at the time of puberty in males. Secondary spermatocytes, both of the secondary spermatocytes divide by meiosis second and form four haploid cells called spermatids. These are haploid cells, and they are converted into spermatozoa by a process called spermiogenesis. Which process? Spermiogenesis. Spermatozoa or the sperm, and these spermatozoa are then released into lumen of seminiferous tubules, and it is called spermiation. The process of releasing mature sperms into lumen of seminiferous tubules is called spermiation. So spermiation, spermiogenesis, and spermatogenesis. Spermatogenesis is the whole process of formation of male gametes. I hope this is all very clear to you. Now we read the text. In males, sperms are produced by spermatogonia, the diploid cells, immature germ cells we can say, which are present in the inner lining or inner wall of seminiferous tubules where they are formed. Spermatogonia increase in number by mitosis, and these are diploid. They divide by mitosis and called primary spermatocytes. They undergo periodically they may undergo meiosis further to form secondary spermatocytes. First meiotic division, two haploid cells and equal cells. Importantly, equal because oogenesis में this division will be unequal, and they form secondary spermatocytes. These further undergo meiosis second to give rise to 4 haploid spermatids. These spermatids are converted into sperms by spermiogenesis. This is transformation, and sperm head gets embedded in the Sertoli cells. Important second type of cell in the seminiferous tubule, that is Sertoli cells. After spermiogenesis, and is released from the seminiferous tubules by the process called spermiation. So spermiation is the release of mature sperms into seminiferous tubules. Spermatogenesis starts at puberty, but oogenesis begins at the fetal stage in the ovary of the female, right? By the action of gonadotrophin releasing hormone GnRH from pituitary. GnRH is from hypothalamus, which in turn causes release of 2 gonadotrophins called LH and FSH from pituitary, anterior pituitary. You might remember from 11th class, human physiology. LH acts on Leydig cells. See, Leydig cells, important. Leydig cells are the interstitial cells located outside the seminiferous tubules inside the testicular lobes. Testicular lobes जो कि 250 एक टेस्टिस में होती हैं, उनमें सेमिनिफेरस ट्यूबल्स के आउटसाइड में इंटरस्टिशियल सेल्स प्रेजेंट होती हैं. दे आर कॉल्ड लीडिक सेल्स, एंड LH काम करता है लीडिक सेल्स पर, एंड इट कॉजेज देम टू रिलीज एंड्रोजन, दैट इज टेस्टोस्टेरोन, एंड दैट एंड्रोजन टेस्टोस्टेरोन stimulates the process of spermatogenesis. And FSH का क्या काम है? FSH act on Sertoli cells जो कि spermiogenesis को start करेगा, stimulate करेगा. Spermiogenesis means conversion of spermatid into mature sperm. Now we see the structure of mature sperm. It has 4 parts, we say head, then neck, then middle piece and tail. Head region at the tip have specialized enzymes filled in acrosome. It consists of enzymes which will help in the dissolving zona pellucida of the ovum or the secondary oocyte while fertilization. Nucleus, red colored. This nucleus consists of haploid number of chromosomes that come from male parent. Behind that there are 2 centrioles in the neck region. Middle piece, centriole will help in division, or say during the fertilization they may contribute. Middle piece has mitochondria, large number of mitochondria, and they will provide energy during movement, and tail obviously help in movement of the sperm in the seminiferous tubules and inside the tract, female reproductive tract. It is sperm cell, structure of mature sperm. Now see the text. Mature sperm consists of 4 parts: head, neck, middle piece and tail. Whole sperm is enclosed in plasma membrane because it is a single cell. So all around it has plasma membrane. Head consists of haploid nucleus and cap-like structure called acrosome. It contains enzymes which help in fertilization to dissolve the zona pellucida of secondary oocyte or ovum. The middle piece consists of several mitochondria which will produce energy for motility of sperms. Sperm released by seminiferous tubules are transported by accessory ducts. Accessory ducts means epididymis, zona efferentia first, then epididymis where they are stored temporarily, then vasa deferens and ejaculatory duct. Secretion of epididymis, vas deferens, seminal vesicle and prostate gland. They are essential for maturation and motility of sperms. It's all about spermatogenesis. Now we come to second process, that is oogenesis, formation of female gamete in the ovary of female. The ovarian stroma or ovarian epithelium, germinal epithelium of ovary has cells called oogonia. These are double set of chromosome or diploid cells. Oogonia undergo mitosis and increase in number. These cells formed by mitosis of oogonia are called primary oocytes. Primary oocytes are being formed when the female is in fetal stage inside her mother's womb or uterus. Primary oocytes, large number of primary oocytes are formed, and primary oocyte start dividing by meiosis first, and during prophase first this division is arrested temporarily till puberty of the female. And at puberty, when the menstruation starts or the menarche begins, this meiosis first again starts and completed to form two unequal cells, secondary oocyte and first polar body. See, this meiosis first is unequal, cytokinesis during this division is unequal, and in spermatogenesis it was equal division. Now secondary oocyte is formed and first polar body is formed. The secondary oocyte then undergo meiosis second. The division or this meiosis second is also get arrested at the metaphase stage, and it remain arrested till then the sperm, one of the sperm get entry into the ovum. The entry of ovum will trigger this division to complete. So after completion of this division, an ovum and second polar body will form. Ovum is converted into, transformed into mature egg cell, and the two polar bodies do not have any role to play, so they get degenerated. Now see this structure of transverse section of ovary. There are primary oocytes. They start surrounded by some follicular cells, one layer. It is called primary follicle. Later on, it may be surrounded by some more layer of cells called secondary follicle, and then mature follicle or Graafian follicle, characterized by antrum cavity. And after that, ovulation occurs. Ovulation occurs at that time the female gamete is in secondary oocyte stage, and when it ruptures, and after ovulation the secondary oocyte, where the meiosis second got arrested at metaphase stage, and entry of sperm will complete this division and ovum will form. And if fertilization does not occur, then it will released out by menstrual flow during secondary oocyte stage only. So see this process once again in the camera. You focus on the camera again. See, oogenesis. Oogonia cells, then mitosis occurs. It is primary, it is not spermatocytes, actually primary oocyte it is primary oocyte. This is oogonia, primary oocyte by mitosis, and this all is happening at fetal stage, and after childbirth, at puberty, this meiosis first get arrested at the prophase first stage, and after birth, when the female reaches the puberty, the division is completed and formed two unequal cells by cytokinesis called secondary oocyte and the first polar body. First polar body get degenerated. This secondary oocyte now divide by meiosis 2. Meiosis 2 also get arrested at metaphase stage, and during meantime the sperm may enter during fertilization. At that time, after entry of sperm nucleus, the final this meiosis second is completed and ovum and second polar body is formed, which also get degenerated. So these are the divisions. Two times the meiosis get arrested, first at the primary oocyte stage and second at the secondary oocyte stage. Meiosis 2 also get arrested, and it get completed due to entry of sperm in the ovum or the secondary oocyte. So during oogenesis also, see the text again. Ovum is formed by the process oogenesis. It starts at embryonic stage growth and millions of gametes, oogonia cells are formed. Oogonium is singular. At fetal ovary, these cells undergo meiosis first but get temporarily arrested at prophase stage and are called primary oocyte. Before reaching puberty, a large number of primary oocytes degenerated, and only 70 to 80,000 left in each ovary. Remaining ones, 70 to 80,000, they get surrounded by one by one. They get surrounded by granulosa cells and form the new theca, or called secondary follicle, or first primary follicle, then more layers of cells, secondary follicle. The secondary follicles then mature into tertiary follicle, and they have a fluid filled cavity called antrum. At this stage it is called Graafian follicle. Primary oocyte present within the tertiary follicle completes meiosis 2, complete meiosis first, and results formation of haploid secondary oocyte and tiny polar body. This polar body get degenerated. The tertiary follicle further changes into Graafian follicle, and inside it there is secondary oocyte. It is surrounded by zona pellucida, a layer, protective layer. Then the Graafian follicle ruptures to release ovum, and it is called ovulation. So during ovulation, the ovum is in secondary oocyte stage. Now see menstrual cycle and fertilization. Menstrual cycle, diagrammatically, hypothalamus release GnRH, that is gonadotropin releasing hormone, which stimulate the anterior pituitary to release two gonadotropins called FSH and LH. FSH causes the growth of follicles and LH causes the ovulation and further maintenance of corpus luteum and release of progesterone from corpus luteum. FSH also release the estrogen hormone, that is female sex hormone, from the follicular cells. Estrogen and progesterone maintains the menstrual cycle. Estrogen maintains the prepare the endometrium, and that is proliferating stage, and progesterone will cause high progesterone will maintain endometrium after fertilization during gestation period. See, menstrual cycle is the reproductive cycle in all primates and begins at puberty, that is menarche stage in female. In human females, menstruation occurs once in 28 to 29 days. The cycle events starting from menstruation till the next stage, next one is called menstrual cycle. During middle of the menstrual cycle, there is ovulation or release of ovum. The cycle starts with menstrual flow that continues to 1 to 5 days or 3 to 5 days. It causes breakdown of the endometrium of uterus. Blood vessels along with some lining of uterus that goes out if the ovum is not fertilized. It is followed by follicular phase. After menstrual phase, second phase is follicular phase. In this phase, primary follicle mature into Graafian follicle. This causes regeneration of endometrium. Endometrium get prepared and primary follicle mature into Graafian follicle. It was second stage follicular phase. First was menstrual phase, second follicular phase. And now it comes, these changes brought about by ovarian and pituitary hormones LH and FSH. They cause follicular growth, and growing follicles produced estrogen. LH and FSH are at their peak in the middle of the cycle, 14th day, and it causes rupture of the follicle and release of ovum. It is called ovulation or the ovulatory phase. The remain of Graafian follicle after ovulation get converted into corpus luteum. Corpus luteum, the important term. It secretes progesterone, the second female sex hormone, for the maintenance of endometrium during implantation and gestation. In the absence of fertilization, if the fertilization does not occur, then the corpus luteum degenerates, get ruptured, and causing disintegration of the endometrium, and again the menstrual cycle begins. In human, the menstrual cycle ceases to operate at the age of 50 years, and it is called menopause. So, today we have discussed the spermatogenesis, oogenesis and menstrual cycle. I hope you could understand the concepts. Read again and listen to the video carefully. Thank you very much.